right, so what is gluconeogenesis? Like literally, what does the name mean?
Creation of glucose.
Creation of glucose. Okay. Everybody agree, disagree? What's neo? New. Okay, so yes. All right. So many tissues, including the brain, depend almost completely on glucose for the metabolic energy. Okay, especially the brain. Does anyone know why?
Some things can't pass the blood-brain barrier.
Some things can't pass the blood-brain barrier. What do some of the other tissues what can they rely on for energy instead of glucose and sugar? What do they get their energy from? Fat. Okay. And is your brain a fatty place? Like, do you have lots of places to store fats in your brain? No. Okay. But no, the brain alone requires if you're healthy, a healthy person, requires about 120 grams of sugar, of glucose, which would be more than half of all the glycogen that you store in your body. Okay, and so we'll talk about that in the sense that, we can't be solely glucose or glycogen based because of the fact that there's no way for us to be able to store enough. And secondly, remember, it also is not fully reduced. It's already partially oxidized, sugar is. And secondly, it carries water along with it. And so therefore, even if we were able to store it, it would be huge, which I'm sure is one of the metabolic reasons why that some of the big old bad guys are so big, since you're talking about your evil, evil ones. All right, but yeah. So anytime that there's during a fast, and even when we say fasting, it could mean between meals, okay? It's not like the overnight fast necessarily that we're talking about if you go into blood work. Or if you've been doing extensive exercise, your amount of glycogen you have available is going to be decreasing, and so therefore you're going to have some way in order to produce sugar. Yes, when I first moved here to Florida, this was the dog that I had. And so she's a little tiny Yorkie, and my girlfriend at that time, just thought that every female in Florida needed a little bikini, and so she bought her a bikini. I did not buy it. So I think I called her Shug Opal, I think wore it like a total of once, maybe twice, and so I can take a picture once for her, but that's all right. But no, she passed away. She got pancreatic cancer. Yes, but no, it's sugars and glucose, and so you got to find some way in order to make that glucose, since we can't break it down from the glycogen like what we talked about in the last lecture. And so that's the process of gluconeogenesis. So gluconeogenesis, what's the starting point for the traditional gluconeogenesis? Pyruvate. And how many pyruvate do you need to start off with? 2. Since, it's going to take, it's got 3 carbons, you're going to need total 6, and blah, 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 blah. So I should say 2. In the sense it's going to take 2 pyruvates in order to make 1 glucose. Let me specify that. It takes 2 pyruvates to make. All right. So, gluconeogenesis is not the exact reversal of glycolysis. You need to know the reasons why, and also how does gluconeogenesis work. So some of the enzymes, they do share with glycolysis, you need to know those, but you need to know the way that they get around it. Remember those 3 steps. There's phosphoenolpyruvate, which is also commonly lovingly referred to as phosphoenolpyruvate as PEP. Come on, you guys need some program. I think in the afternoon. But so PEP to pyruvate, okay, was one of the checkpoints for glycolysis. And what enzyme did that one? PEP to pyruvate an ATP. What was that enzyme in glycolysis?
Was it enolase, or is it a dehydrogenase, or
It's what?
is it kinase?
It's pyruvate kinase. All right. And so then we also have to get around fructose 6-phosphate, fructose 1,6-bisphosphate. So what step was that one? What enzyme did that in glycolysis? That was the second checkpoint.
What enzyme did that?
Mm-hmm.
Was it the
Your BFF? No. PFK1, yes, phosphokinase 1. It was not to be confused with PF chains. Okay, and then you also have to have a way to get around glucose to glucose 6-phosphate, which is hexokinase. Okay. The net result is going to be the reversal of these, but you have to know the different reactions that they use and what enzymes that use them. Okay, some other things to keep in mind is where does gluconeogenesis take place? Okay. Where does glycolysis take place? We're going to be reviewing this kind of stuff all semester long because compartmentalization is actually a really important, biomedical issue or topic in vivo. Okay. Is it exactly where I counted? The fact that it's not an exact reversal glycolysis. And then what are the feeder pathways? So we have the glycolytic ones and there's the gluconeogenic ones too as well. Like other ways to get the components in order to go through gluconeogenesis. And eventually, which I think technically is next chapter. Like I said, sometimes I forget where chapter 14 ends and 15 begins. We also talk about the hormonal input and for glycolysis and gluconeogenesis. All right, this is just a review here of the glycolytic reactions. Okay, just to show you, again, those that are highlighted are steps 1, 3, and 10, which are the 3 checkpoints, which cannot be the reversal for gluconeogenesis. The others you can. So PEP, which makes sense when we talk about PEP, remember it's high energy, so it should make sense that it can go, once you make it can go backwards. And all those others can go backwards except for steps 1, 3, and 10 of glycolysis. So let's get started. Once again, this was part 3 of the PowerPoint. It is available on my fire. You got to think about this enzyme. So let's say that I'm drawing, you know how glycolysis kind of looked, I kept referring to it as linear, so this was glycolysis. Well, this very first step, going backwards, which would have been step 10, it actually takes more than one step in gluconeogenesis. Okay, so that's what we're looking at. We're looking at the first step to circumvent that in-section multi-step process. Which should make sense. Remember, PEP is really, really high energy. Okay, so in this one we have pyruvate, you have ATP, you have carbon dioxide, you have water. You got a lot going on here. Goes on to make oxaloacetate. Oxaloacetate, a lot of times I abbreviate it as OAA. I think maybe what you abbreviate as OAA. It's going to be your friend. Remember there are those common metabolites, pyruvate by the way is one of them, DHAP and glyceraldehyde phosphate, and oxaloacetate, OAA, is a biggie. Okay, so this is your first time, your first introduction to OAA. Hello, OAA, how are you? And so, does anyone want to venture, what other pathways do you see OAA? It is, it's part of the citric acid cycle. Mm-hmm. Does anyone know what else? What else is OAA used for? What kind of compound is OAA, if we look at it? It's got carboxylic acids, it's got ketones, so if you put those together, it's called a
carboxyketone.
Close. A keto acid.
Okay.
And what's the position of the ketone to the carboxylic acid? So we talked about this just 1 or 2 classes back. What's the positioning? For the what kind of keto acid is this? This position is Matthew Scales. It's not Matthew Scales, but you could say it's position 2, but it would be alpha. This is an alpha keto acid, which sounds an awful lot like what kind of alpha blank acids that we talked about. Connect the dots here. Connect the dots. You gotta start connecting them. What other kind of acids, blank acids, alpha blank acids are there?
Amino acids?
Amino acids. Thank you. So this is another ketone. Remember, what is pyruvate? Alpha keto acid form. There's pyruvate and it becomes, so it's the keto acid for what amino acid? Alanine. This is the keto acid for an amino acid. What amino acid would that come from? No. Asparagine? No. Aspartate or aspartic acid. And so this is actually also a way to use to convert aminotransferase. In particular, aspartate aminotransferase will convert between acetylacetate and aspartate. Okay. And so we'll see that over and over again as well. All right. So this is the first step. It does have a different cofactor, which we'll talk about in a moment. And that's the enzyme name. It's also under allosteric control. It's activated by acetyl-CoA. Does anyone know what is acetyl-CoA? Notice there is no acetyl-CoA here, so it may not be so obvious. But where do we get acetyl-CoA? Why would it ever accumulate? Under what conditions? Where does acetyl-CoA? This is another common metabolite you see a lot of. Where does it come from? What?
You need to get into the citric acid cycle, right?
It goes into the citric acid cycle.
Okay. Well, would it be with
But where does it
low
come from?
energies? Because,
If you have low energy, you can have a lot of acetyl-CoA.
well, I'm assuming, if you're making glucose,
Okay. If you have a lot of energy, you're going to break down glucose? No.
okay?
Right, because that means you don't need it anymore, right? So gluconeogenesis in general, another time that you undergo gluconeogenesis is when you have a lot of energy or when you have not enough energy. What are some of the reasons why you undergo gluconeogenesis? If you've already got a lot of excess energy around, then you're going to want to store. Your body goes into, that's one of the reasons why, be careful, so your body will try to start to store glycogen. And so it'll go in reverse. Okay, but where else? Where do we get the acetyl-CoA? Something from that. That's the byproduct of another metabolic pathway. When you break down a certain biomolecule, acetyl-CoA is the product. I haven't covered it yet, that pathway yet, but where do we get it? It's not sugars. It's not proteins. It's fat. Okay, so it's your fatty acids. So it comes off of fatty acid catabolism. All right, which makes sense. You got too much around. It's breaking down the fats, and that way you can. So in that sense, you're gonna be using it to help turn this on. Okay, either to store glucose, or in form of glycogen, and so on and so forth. Okay, so what class or classes of enzymes can this be? Transferase. Okay. What are you transferring?
CO2 group,
CO2 group, and that's called a.
carbon dioxide.
Well, that's the molecule, but whenever it's a functional group, it's called a carboxylate. Okay. And so this would be a carboxylase, which is a specific. You can say carboxyl transferase, but they commonly call it a carboxylase. So if you said either one, I would accept that one. But yeah, they call this one carboxylase. What carboxylase is it? What specific one? It's pyruvate carboxylase. Okay, so pyruvate carboxylase requires a lot, because there's a lot that's going on here, right? It looks at what, if you remember, it looks like it's one step. In reality, it's multiple steps by one enzyme. And so one thing, remember, what's one thing that you should already be able to guess it's going to need, knowing that it's got these carboxylate groups and it's got ATP, what pretty much almost always comes along with those? What's the other cofactor that will always come along with those? Magnesium. So magnesium 2+ is required, but it actually also requires biotin. Okay, and I've already mentioned this a little. Okay, so biotin is a cofactor that you need to know about. It's involved a lot of times with these carboxylases. the transfer of carboxyl groups. This is pyruvate carboxylase. Okay, and so what does biotin do? I don't know why it's on there twice, but okay. Remember, we still actually haven't made, like if I were, I don't have it on here, and I can't, unfortunately on the Mac, I can't write on multiple slides without carrying it over from time to time. So, this is the reversal. So we had pyruvate. In glycolysis, pet to pyruvate was pyruvate kinase, and it was just one step. Here we've only made OAA. I shouldn't say only. We made OAA. We've got to go from OAA to PEP, ultimately. So it's 2 steps to go circumvent that one. Pyruvate carboxylase is the very first step. But the role that what biotin does, it looks weird, but it transfers carboxyl groups. It is a cofactor. And it has, this is where the ATP comes into play. Biotins get attached to lysine groups. So the enzyme will have a lysine on it, which has an activated nitrogen, so the biotin forms an amide with it. But it's the other end. In the other end, that nitrogen can transfer down that carboxyl group into the right orientation. And the way that that happens is actually something that should have seen in organic chemistry too. Because remember, pyruvate is a keto acid, but the one that it is in particular is it's got that methyl group next to the ketone, and so it can form the enolate. And so it's this, that enolate addition, I think it's got a special name, I can't think of it off the top of my head. But where the enolate comes down, boom, side effects optional, and then you swing out the alpha to the alkene to attack that carboxylic acid, the carboxylate off of the biotin. Okay, so that's how we get OAA. OAA is another one that you're probably going to know the structure for it, or you should know the structure for it, hint, hint, by the end of the semester, probably by the end of chapter, was at 16, just because of the fact that it looks just like aspartate, except you remove the amino group and you get the keto form. And we're going to see that a lot. Alanine and pyruvate, I've already mentioned, OAA and aspartate, and then we'll talk about glutamates, alpha-ketone glutamate. And those are 3 biggies, but they're not mutually exclusive. Okay. This is step 2. And unfortunately, step 2's got 2 different names. But they both kind of make sense. I like the animal name the best. Oh, one of them's in animals, one of them's in plants. I had to look at that one time, like why. And I don't know historically why they call them 2 different things. But in this one, we take OAA. What's one thing that's different about this? To make PEP, what's the B thing? Just anything. State one thing that you can notice. What? It uses GTP. You have GTP. We get GDP. We need to edit a phosphate. I think this is the first time that we've seen GTP used this way. Okay. Also, what else happens in this? So we edit the phosphate. What else is different or is occurring in this? Pardon?
A decarboxylate.
You're decarboxylating something. You're removing that carboxyl group. Okay. So what are some of the classes of enzymes that can do this? Like a decarboxylase or a carboxylase sometimes. What's another one? What can do some type of function like this? That we talk about with GTP, phosphates, and GDP. It's a kinase as well. So if we put that all together, we get PEP carboxykinase in animals. That's what we call it. PEP carboxykinase. Because it's between both. To make PEP. So plants call it PEP carboxylase. They just forget the kinase part. The animal name helps you remember that that's possible. Like I said, I don't know historically why there's a difference. I'm sure there's a reason. So, we had pyruvate, I don't know, carboxylase, I'm abbreviating these. Then we had PEP, carb, Okay. In order to make PEP. So if we just look at the general, well, why not you can get, this is just the abbreviated version for pyruvate carboxylase in the book. So once again, use biotin, ATP, ADP. It doesn't list magnesium, but it does require magnesium. And then the second part, I like this one because it shows you the mechanism. This is something similar to what you've seen or should have seen in organic chemistry too. Yeah.
Biotin's only used to add carboxyl groups.
Well, in the transfer carboxyl groups, I guess. Remember, in entomology, many times some of these will go both directions, but no, we usually just say about the transfer carboxyl groups.
So it wouldn't be used here?
Pardon?
It wouldn't be used here?
It's not used in this instance, no. It's not used every time you carboxyl group. It's one of the things they have in your tool belt. Those that are evolutionary biologists would say that it didn't evolve to use biotin, that this one didn't evolve to use biotin. It was designed to use biotin. You don't have to. In this instance, you actually don't really need it because if you remember from organic chemistry, this is oxalic acetate is really special. Okay. Because to my knowledge, it's the only biomolecule that this is true for. It has to be true. It's an alpha keto acid, and alpha keto acids have their own special properties, but it's also a beta keto acid. And so if you think back to organic chemistry 2, decarboxylation occurs for beta keto acids, or can occur in beta keto acids. And this is just a beta keto acid decarboxylation reaction from organic chemistry. And so you get the collapse of that, this breaks, which would form what would be an enolate, so it's sort of like the enolate attacking. Well, actually it is an enolate attack, so yeah, it's just one. What the enzyme is going to do is going to help get everything in the right orientation and to speed that up. Okay, and so that's why it's carboxykinase. Right. Any other questions before I show you the next slide? Okay. So these are some things to consider, and you really have to, like I said, start to put things together. It's a little bit more complicated than glycolysis in that sense. So where does glycolysis occur? So where is the pyruvate in glycolysis? Where does gluconeogenesis occur? Especially the first steps here, gluconeogenesis. The fact that there are two paths ultimately for pyruvate to be transformed into PEP. We'll talk a little bit about what all this means in a moment. Where does glycolysis occur? That's why I've been trying to say this over and over and over and over and over again. Glycolysis, and later in the semester, we'll be drawing our cell. Don't hate me for my ability to draw. And especially there's the cell, we've got the mitochondria, it's the outer membrane, inner membrane space, mitochondrial matrix. You may have the nucleus in here later on, and so on and so forth. So the nucleus, lysosomes, whatever you want to put in there. Okay, so where does glycolysis occur? It's out here. Okay, so this is glycolysis. So therefore, where's the pyruvate in glycolysis? It's in the cytosol. Okay. Where does gluconeogenesis begin? Pardon? It is not cytosolic in the beginning. No, and it's close, but no. Okay, it's not in the outer mitochondrial membrane. And so, but what happens, and there's a reason, and I think that's technically chapter 15, which I would kind of like to put in here if I was writing this, is you can imagine one nice thing about keeping them separate is if you didn't have ways to control it, the moment glycolysis would make the pyruvate, if it was occurring right here, gluconeogenesis would just go ahead and convert back, and it'd be called a futile cycle. And so this is by keeping gluconeogenesis beginning down here in the mitochondrial matrix, then it's a way to at least get it started someplace else so that way they're not directly fighting against each other. Okay, and then the paths for pyruvate depends on the availability of lactate. We've already discussed where lactate comes from. Cytosolic pyruvate, so how much you have around, and alanine. Why does it make sense that alanine is going to play a part in the role of pyruvate?
Because it's the amino acid.
It's the amino acid version of pyruvate, the counterpart. Pyruvate is its alpha keto acid version. You want to think of that. So if you have a lot of alanine, you can make pyruvate. If you need alanine, you could use up pyruvate. And so, and a lot of it's also going to depend upon the cytosolic need for NADH. NADH is very important. Okay. One thing, and I don't know that. So one thing is NADH. There's a cytosolic pool of NAD, NADH, and a mitochondrial pool of NAD, NADH. The two shall never meet. Okay, so don't ask me how they separated out and everything, but they do, okay? In the beginning, God spoke and they did, okay? But there are these two pools. You keep them at check, but they cannot pass. It's much like Moses with the Holy Land. A lot of things cannot get to that Holy of Holies, also known as mitochondrial matrix in cell biology. So, and NADH is one of them. There are ways around it. But it doesn't directly. And so that's why it depends on the need for cytosolic NADH versus mitochondrial. Okay, so first method. We don't have time. Good. First method is whenever pyruvate is converted to OAA, it has to be transformed to malate. So we were showing it right here, pyruvate to OAA directly to PEP. That's gluconeogenesis. However, pyruvate and OAA don't necessarily mean that it's going to make cytosolic PEP, okay? Because OAA typically is going to be mitochondrial. That's where the citric acid cycle occurs. Hint, hint, that's going to be coming up. We're going to be drawing that into here. This is not drawn to scale, I would say that, because the mitochondria is really tiny, but I'll just pull it up. Okay, because the mitochondria have no transporters for OAA. You make it inside here, it can't just jump across all the membranes in order to get to the cytosol in order to make PEP. You have to have a way around it. What it does have is malate. Malate is another metabolite that we'll see multiple times. What's another metabolic pathway that has malate in it? It's the citric acid cycle. So in reality, gluconeogenesis and the citric acid cycle intersect. Okay, that's not on a scale either. Okay, and so we're going to see that a lot of these cycles are not, metabolic pathways are not mutually exclusive. Just like glycolysis and gluconeogenesis share like 7 steps, gluconeogenesis and citric acid cycle indirectly affect each other as well. Second method is we have alanine. Let's say you really need to get that pyruvate fast. We can't make it fast enough inside the mitochondria, but we have alanine around, okay? And so what enzyme can convert the alanine into the pyruvate? What specific enzyme? This is when you would want to draw out, sound effects optional. Oh, don't draw that. I was using the expo marker on the iPad. Don't do that. Okay. So if you draw alanine, alanine looks like so. What's the, oh, there's a carbon right there. What's the R group for alanine? Methyl. I'm going to go ahead and draw it out. Okay. Pyruvate looks the same thing, except instead of having the amino group there, what does pyruvate have? It's an alpha keto acid, so what does it have instead?
Carbonyl.
It's a carbonyl. And this enzyme is definitely, I'm showing it as one step in reality. The mechanism itself has more than one step. It's definitely reversible. Oh, I forgot my negative. A negative charge. What enzyme does that? What kinds of enzymes can do it? Are we carboxylating something? How many carboxyl groups are on alanine? carboxyl groups? How many are on alanine?
One.
One. How many carboxyl groups are on pyruvate? One, so it's not carboxylase. Oxidase or reductase? Technically, we aren't oxidizing the carbon. Go back to, if you go all the way back to the redox numbers, carbon and nitrogen's still more or less negative than oxygen, so therefore, that's not it, is what I'm trying to say. What else is?
Is it really one enzyme that does it?
Yeah.
Switches two groups out. I mean, what is that?
Switches two groups out.
Switches the amino or carbonyl group.
Oh, what was it? It's a transferase. What transferase is it? It's aminotransferase. What specific aminotransferase is it?
Alloaminotransferase.
What?
Alloaminotransferase.
No. Close. You were really close. Not al. Alanine aminotransferase. This is alanine aminotransferase.
Well, I was
Yes.
just combining the words.
Yeah, it's very new speak for you. Okay, so this is alanine aminotransferase. Because you say it's transferring it out, what it is going in the reverse direction. The amino group gets transferred into water. That's where the oxygen comes from water. So it's why it's not considered a redox reaction in that sense. You know, this is alanine aminotransferase. And so that's the enzyme. Come on now, let me do this. Oh, it's going to get there. For the second method. The third method is if you have a lot of lactate around under anaerobic conditions, remember it can also be transformed into pyruvate. Okay. And we already talked about that with lactate dehydrogenase. So that's nothing new. But yeah, so those are the three ways. Either you're going to use the OAA, but the OAA has got to be transformed to malate. Malate can cross back over. So this is the enzyme that we'll talk about in a moment that does that. There's a cytosolic version and there's a mitochondrial version for it. That happens a lot. Okay, a lot. A lot of the enzymes that are inside the mitochondria may have counterparts out there that if they do the same type of reactions. If there's an isoform that stays in the mitochondrium, there's one that stays out in the cytosol, never the two shall meet, that kind of thing. Okay, so here's this little picture that kind of explains all of this. Explains this. I like this picture. It's almost as pretty as my little cell. Okay. I don't know where you want me to start. So this is method 1. It doesn't show method 2, but this is method 1. You have the pyruvate. The pyruvate from glycolysis would have to be transported in. There's a transporter for pyruvate. It comes into the mitochondrial matrix. Inside there is where pyruvate carboxylase occurs. I should have hidden this one. That way you could have told me the name. I would expect you to have been able to come up with the name for this one. It's malate dehydrogenase because it takes an NADH to an NAD and it transforms oxaloacetate to malate. Malate comes out there and the cytosolic version just goes ahead and does the opposite. So this is effectively the way to get NADH to the cytosol. You see that? Without the NADH itself actually leaving. There's a pool, as I mentioned before, there's a pool inside the mitochondrion, there's a pool inside the cytosol, never the two shall meet. This is the I should say cheating, because this is the actual way that we do it. This is the way kind of short circuit that, because we can essentially take the electrons from the NADH in the mitochondria and transfer them to the NAD plus in the cytosol to make NADH if you need the NADH out there. We're going to find out this is reversible, by the way. It's where it's also a way that you can move the NADH into here. Okay. And then, well, obviously it's reversible because we did the reverse here. The cytosolic malate dehydrogenase makes the NADH. You get your OAA, ultimately make PEP. PEP then goes on to continue on with gluconeogenesis. Another way is under anaerobic conditions, or if you have a lot of lactate, you use lactate dehydrogenase, which we talked about like early on, and the pyruvate can move on through here. And then once again, we undergo through all of this, then the PEP can go ahead and can leave. Okay. Whew. Control mechanisms. This is called the Cori Cycle. This is one way to get the lactate back. I haven't double checked for the most recent list, but I know that it used to be listed specifically on the MCAT to know. No, if you're working out, you're feeling the burn, you're literally feeling the burn. Okay, the lactic acid is going to be building up. You would short circuit and most likely die. If you didn't have a way in order to cycle it back, you're going to utilize the Cori cycle. So this is one way to convert it back ultimately to glucose. So, I'm actually going to, well, that's just the word I cannot speak today. That's just the word way of saying it. You know what I mean, I think. Okay, moving on. This is the picture. This is the cartoon. Sometimes, at least for me, it's easier to visualize, even though this is from the older book. Okay. So, and I don't know why they picked your leg, but I'm not pulling your leg. Bada bing. I'll be here all next week. Okay, so they're showing the leg muscles for skeletal muscles, you know, under anaerobic conditions. You know, you've been working out, you've been using all the glucose, it goes to pyruvate, but there's not enough time in order for it to go under aerobic conditions in order to regenerate, so it goes to lactate. Lactate will build up, okay? What it does is it releases it through the blood system, which will travel to the liver. Once in the liver, lactate dehydrogenase reverses, and get the pyruvate. You can go up through gluconeogenesis to get the glucose. The glucose can then be shuttled back to the blood from the liver to go back to the skeletal muscles, and so that's why it's called the Cori cycle. Okay. Any questions on the Cori cycle? Cori cycle, not Cori. Cori cycle. I really can't speak today. That's because I need a break. It's Friday. Okay, so back to this figure on the board. So we made PEP. It goes through the reverse of glycolysis until you got to what would be step 3 of gluconeogenesis, which was phosphofructokinase 1. And so therefore, you've got to have a way to get around it. I like this enzyme. I like a lot of these enzymes. But this one is so much more simple compared to what we had to get to PEP. Okay, we have fructose 1,6-bisphosphate. We're just going to try and make fructose 6-phosphate and give off an inorganic phosphate. On the reverse, it required ATP, and that's why it's called PFK, or phosphofructokinase 1. All right. What class or classes of enzymes can do this? This. It's a phosphatase. Okay? It's a phosphatase. So now what's being phosphatased? My old boss hated it when we would take words like that and make them our own verbs. But I'm like, you know exactly, if I say it's phosphatase, you know what I mean. It's really clever. Okay, so what exactly is being phosphatased? What? Right, but no, I mean, what's the name of the compound? Fructose 1,6-bisphosphate. So now is the time for you to do your little newspeak. You guys know what I mean when I say newspeak? No. You guys ever have to read 1984 by George Orwell? Mm-hmm. Okay. Well, it's a great book. And it's a great political book, and some would say theology and ideology and everything like that, but what they do is the idea is they take words and they want you to learn to speak in as few, they say language is a way of expression, and they want to limit expression, so they to use as few words as possible. So it's called newspeak, it's a different language, and so they put words together. But if you put these together, fructose 1,6-bisphosphate and phosphatase, you wouldn't want to say fructose 1,6-bisphosphate phosphatase, but it would become fructose 1,6-bisphosphate, yes. See, I told you, that makes perfectly logical sense. I like this enzyme. So perfectly logical sense in the sense that it's phosphatasing fructose 1,6-bisphosphate. Reaction number 2, using glycolysis wording, would be fine. You know, it's reversible. But reaction number 1, which was hexokinase, is not. Okay. No, yeah, we already said that was using hexokinase. So we have to have a way around the hexokinase. Oh, I'm sorry. I think that part of my words got chopped off on this one. I apologize if that. When it, yeah, that's supposed to say glucose 6-phosphate. Well, whenever I converted it, it didn't. The phosphate got. That's supposed to be a phosphate right here. Whoa, sorry. That's supposed to be phosphate. Okay. So we have glucose 6-phosphate. We're making glucose. We get off in inorganic phosphates. What class or classes of enzymes can do this? What class of enzymes will do this? It's a phosphatase. We're phosphatasing. What are you phosphatasing? Okay. Can you speak it? Oh, see, you're on fire. What? If you had to guess, if I said there is a cofactors required for this to occur, what would it be? It's magnesium. Once again, that's just a default. You should see, anytime we have phosphates and stuff, you should just always write down. I will give you a hint. This next slide is important. Asterisks. Especially for those of you who are wanting to go in biomedical fields. This is the kind of logic and stuff that we should start to think about. Okay. It's actually a magnesium. This is where I want you to start to think about it. It's very specific. You only have this enzyme on the luminal side of the ER in hepatocytes, which where are hepatocytes at? The liver. Renal cells, which where are those? The kidney and epithelial cells of the small intestines, which is in the small intestine. Okay, other tissues therefore cannot supply glucose to the blood. Gluconeogenesis. Now they can do the lactate and all that kind of stuff. Why is it important that you only have glucose 6-phosphatase on those specific places? So just think about what happens in those cells. Why would you not want to do this? Like, for example, why is it not in the brain? Oh, I should have looked at the verse in the Bible there in the New Testament about the different roles of the body and all that kind of stuff. This is good. Like in the biochemistry version.
Is it for filtering?
No. Even though, I mean, filtering does occur in these areas, but that's not the specific reasons here. What is the goal? Let's say the brain had this enzyme. So glycolysis kicks off in the brain. The brain needs energy from glucose. It makes glucose 6-phosphate. What would happen if it had this enzyme? Well, it would just take that glucose 6-phosphate and make it back to glucose. Just round, and you would die. Okay. Okay. So, therefore, it prevents a futile cycle from happening. Only these cells can initiate, or not initiate, can make the end, terminate gluconeogenesis. Okay. That also means that any glucose produced by the liver or the kidney can then enter the broad bloodstream to provide, or the small intestines. I don't know why I've got small intestines there. They can go on to provide. They're not going to fight with the other enzymes, with the brain, and with the other tissues, with the brain in particular, in order, or the heart, or something like that, in order to get the glucose that it needs, or the skeletal muscles. they're going to say, we're going to hold off right now. We don't need the glucose you do, so therefore we can, I'm really anthropomorphizing these cells, but therefore we can give you the glucose. Okay, whereas if the brain and the skeletal muscle had this in there, the moment that it would go make glucose 6-phosphate, it would just take it back out and make glucose again. So you couldn't go on with glycolysis to occur. Okay. This is just this will be available. Send me an email. I'll put this on. This is especially if I get through this. Overall, this is just the overall, like little cartoony way of showing glycolysis. This is gluconeogenesis to show you the bypaths. So on your own, you can just look over that. I just included that. That way it'd be in your PowerPoint. Then we've already talked about this. There are amino acids that are easy to help. And we're going to see this over and over again. Don't memorize this. Right now, I wouldn't expect you to understand why alanine makes sense and why aspartate makes sense. And it's because they're aminotransferases. And by the way, it's aspartate aminotransferase, these specific enzymes that make oxaloacetate. Alanine aminotransferase is this specific one. At this point in time, I wouldn't ask you, okay, explain to me histidine. Okay, whoa, no. Okay, that's by the end of the semester. But that's just to let you know that it does exist. Once again, this right here, this is just a little cartoon way of showing all of it. Okay. And the differences between those. Okay. I only have like a minute left on this one. Pentose phosphate pathway is always like the redheaded stepchild of this chapter and in general. I always ran out. On your own, please read over all of this. I can understand. This is another way, another pathway. It's kind of like a little offshoot. You want to think of it, for glycolysis and gluconeogenesis. I think they put it in this chapter because of the fact that it has no pentoses or sugars. But it is a little different way. So we're only going to see this. This is the one that kind of made looks like the lattice work with little shoestrings. So when you look at it, and I'll show you what I mean about it in a moment. It's ways to make nucleotides. It's a main way to make NADPH, NADP to NADPH, because we only have a couple ways to do that, and this is one way to do that. Okay. Well, this is just the beginning part. But see, that's why I call it, it's not linear, it's not cyclical, it's like a little lattice work, ultimately. So you first have to make the ribulose 5-phosphate, and then they just swap. All these enzymes here just swap partners, so to speak, and move it from one place to another, until you ultimately get down here, which then can enter into glycolysis, or gluconeogenesis if needed. It's not a little option for this. But you can use this to ultimately go on to make nucleic acids and to form NADPH. So those are the two main ways. So you want to look at it just a little bit those last 2 or 3 slides. And once again, if I don't, like later in labs, let me know and I'll upload it to my fire. The one that has all the answers and all the things to it. Okay, so